Hello everyone, my name is Julia Hetzel, and today I will be discussing the cross-bridge cycle. Before we dive into the cross-bridge cycle, there are a few key ideas and words we will be covering that are important to remember when discussing the cross-bridge cycle. We will be defining and talking about the importance of cross-bridges, adenosine diphosphate, inorganic phosphate, and the power stroke. The understanding of the cross-bridge cycle begins with being able to understand the structure of skeletal muscles, the filaments behind muscle contraction, the sliding filament model, the power stroke, and finally being able to put all of that knowledge together to form the cross-bridge cycle. With all of that said, let's dive in. We will first talk about the structure of skeletal muscles. On the following slide, we will be discussing the structure of skeletal muscles and what makes up a sarcomere. The structure of a skeletal muscle. We will be looking first to the left of the slide where the diagram of the skeletal muscle is located. Skeletal muscles are connected by tendons to bones of our skeleton. In the top portion of the diagram, we will see the fascicles located within the skeletal muscle. Within each skeletal muscle are many fascicles held in by the epimysium. Looking now at the second part of the diagram containing the myofibers within each fascicle, these myofibers are surrounded by the paramysium and the endomysium. On each of the myofibers are satellite cells. Taking a look at the third part of the diagram, we can see the myofibrils and satellite cells located within each myofiber. Myofibrils are surrounded by the sarcoplasmic reticulum. And finally, we get to the bottom of the diagram and are able to see the myofibrils. Under the sarcoplasmic reticulum is where the root of all muscle contraction lies. This is where we will find the sarcomeres. The sarcomeres contain the important filaments actin and myosin. Actin is the thin filament and myosin is the thick filament. Now, we will be taking a deeper dive into the structure of myofibrils and more specifically the sarcomeres. Within the sarcomere, we can find the Z-disc that forms the lateral borders between sarcomeres. There's also the H-zone that contains only thick filaments, the I-band that contains only the thin filament, and the A-band that runs the length of the thick filaments but also contains both thick and thin filaments. With our understanding of the structure of skeletal muscles, we can now begin to look at the filaments that make muscle contraction possible and why they are able to cause muscle contraction. On this next slide, we will be diving into the structures of actin and myosin and building up the foundation of what causes the cross-bridge cycle. The following slide is the filaments behind muscle contraction. This is where we will be looking into what actin and myosin actually are. The structures of actin and myosin filaments and their importance. On this slide here, we have the structures of actin and myosin filaments and their important roles in the cross-bridge cycle. Looking to the left of the slide, we see the thin filament actin. Now we can break down actin and what makes it so important. There are actin molecules that form a chain of spheres. Lying over the myosin binding sites of the actin molecules will be the tropomyosin. We can also see the troponin. Troponin is extremely important. Calcium ions released by the sarcoplasmic reticulum bind to the troponin, and the troponin is able to pull back the tropomyosin to reveal the myosin binding sites. Looking to the right of the slide now, we are able to see myosin molecules, which includes the myosin tail, actin binding sites, ATP binding sites, the two heads, as well as the flexible hinge region. At the center of the slide, we will see these two molecules together. The thick filament is made up of thousands of myosin molecules, with the heads neatly laying together to help move the actin along as the two filaments slide past each other. Above and below the myosin filament, we can see the actin filament. On the following slide, we will be discussing the cross bridge of the cross bridge cycle and discussing what makes it a cross bridge and gives the cross bridge cycle the name, the cross bridge cycle. The cross bridge of the cross bridge cycle. What a cross bridge looks like. Here we see a diagram of what a cross bridge looks like. This can be seen to the left of the slide and towards the bottom right of the slide. Cross bridges occur when the myosin heads are attached to the actin via myosin binding sites. In order for this to happen, two things need to be attached to the myosin heads. These two things are adenosine diphosphate, ADP, and an inorganic phosphate molecule, as well as the myosin binding site of the actin to the myosin head. This is what brings about a cross bridge and gives the cross bridge cycle its name. Now that we have an understanding of what a cross bridge is, let's see how it can be used within our body to aid in muscle contractions. On the next slide, we will be discussing the sliding filament model. What causes the sliding filament model, what moves within the sliding filament model versus what doesn't, and its key role in muscle contraction within our body. The sliding filament model. To the left-hand side of our slide, we can see two illustrations of what the sliding filament model may look like. Now the upper left-hand side depiction is colored, and we can see the myosin filament illustrated in purple. That is going to be our thick filament. And we can see the thin filament actin illustrated in green. Now below that is going to be a much less detailed illustration of the sliding filament model, and we can see to the right hand side of our slide towards the bottom an animation of the sliding filament model in action. In order for a muscle to contract, the sarcomere must shorten in response to a signal from the nervous system. The thick and thin filaments will slide past each other in order to shorten the sarcomere. Many sarcomeres are also shortened as well, which results in the muscle contracting. The process of filaments sliding past each other in order to shorten the sarcomere is known as the sliding filament model. Each sarcomere has filaments that slide past each other when the muscle contracts or relaxes. How they slide is what we will be discussing, and muscle contraction is only possible by these filaments sliding back and forth. We previously saw the breakdown of exactly where these filaments are and how many are required for a muscle to be able to contract. Now, let's take a look into what makes this sliding filament model actually possible. On the next slide, we will be talking about the power stroke and the steps it takes to actually be completed, and why it is so vital to the sliding filament model. The power stroke. It's as simple as 1, 2, 3. The sliding filament model is made possible by the power stroke. The power stroke begins with a cross bridge formation as the ADP, adenosine diphosphate, and inorganic phosphate are still bound with the myosin. Once the ADP and phosphate are released, this causes the myosin to move forward towards the M line. This motion of the myosin head is what pulls the actin filament along. This is the motion that we see in a sliding filament model and how that model is made possible. Now on the next slide, we will be putting all of this information together and we will be discussing the cross-bridge cycle. On the next slide, we'll be discussing the cross-bridge cycle. The cross-bridge cycle. The cross-bridge cycle is just that. It's a cycle. The following steps are constantly being repeated in order to actually contract our muscles. The muscle contracts when these filaments slide past each other, shortening the sarcomere, and then the muscle allowing for muscle contraction. Muscle contraction is initiated by nerve impulses that lead to the release of calcium ions. The troponin of the actin filaments are bound with the calcium ions. This is what moves the tropomyosin, revealing the actin binding sites. The head of each myosin filament at this point is currently bound to an adenosine diphosphate molecule and a phosphate molecule that are remaining on the heads of the myosin after a previous muscle contraction has been made. The myosin heads release the phosphate molecules and bind to the actin. The myosin heads then pull the actin filament by pivoting on the hinge region, just below the two heads. As the heads move, they release the ADP. They stop moving once the ATP, adenosine triphosphate, binds to the head and triggers the myosin heads to be released from the actin. Once this occurs, the ATP, adenosine triphosphate, gets broken down into ADP, adenosine diphosphate, and the inorganic phosphate molecule we saw previously. The cycle then repeats over and over until the sarcomere is fully contracted. Now these steps can be seen towards the center of our slide in a much more simply explained way, starting with step 1, where the myosin binding sites are exposed as the calcium ions bind to the troponin. Step 2, the cross bridge between actin and myosin is formed. Step 3, the ADP, adenosine diphosphate, and inorganic phosphate are released in the power stroke. Step 4, the cross bridge is broken and ATP, adenosine triphosphate, binds to the myosin head. And finally, step 5, the head is returned to its starting point or cocked back when the ATP is broken down into ADP and an inorganic phosphate. And these steps, again, are just repeated over and over to result in the filaments sliding together and allowing for the muscle to contract. And this happens over thousands of sarcomeres within each muscle fiber, and we result in, again, just a muscle contraction. Now we can see this step by step to the left-hand side of our slide, as well as animated just to the right of that. When a person dies, the cross-bridge cycle continues until all the energy, ATP, in their body is all used up. With the lack of ATP within the body, the myosin heads never are able to detach from the actin binding sites. This causes the muscles to never be able to relax after contracting, causing the rigor we see within rigor mortis. Now we've got a pretty good understanding of the previous slides and all the knowledge they had to offer. We will have a knowledge check. This is a totally optional quiz with just 4 questions and the answers on the next slide. We'll begin with question 1. The sarcomere is a part of which muscle structure? A, the fascicle, B, myofiber, C, the myofibril, or D, the sarcomere? Question 2. Which filament is it that contains the binding site for calcium ions? Is it A, actin, or B, myosin? Question 3. Which of the 3 steps of the power stroke are the ADP, adenosine diphosphate, and the inorganic phosphate are released? Is that step 1, A, B, step 2, or C, step 3? Question 4. The myosin binding sites are exposed as the calcium ions bind to troponin. Is what step in the cross-bridge cycle? Is it A, step 1, B, step 2, C, step 3, D, step 4, or E, step 5? Now come the answers. To answer question 1, it is C, myofibril. The answer to question 2, which filament is it that contains the binding site for calcium ions? It is A, actin. And question 3, which of the 3 steps of the power stroke is it when ADP and phosphate are released? The answer is B, step 2. Question 4, the myosin binding sites are exposed as calcium ions bind to troponin is what step in the cross-bridge cycle? It is A, step 1. Now we just have some fun facts about muscles within our body. The first fact is the muscles within the heart, diaphragm, and eyes are constantly working. The heart muscles and diaphragm constantly work around the clock to keep us alive. The next one is the human body contains more than 600 muscles that together make up more than 40% of a person's body weight. The third fact is the heart pumps roughly 2,500 gallons of blood throughout the entire human body daily. And finally, the smallest muscles are located within our inner ear. Now that we have a full understanding of the cross-bridge cycle, we also have a clear understanding of how and why our muscles are able to contract to allow for skeletal movement. And we also have some fun facts to share about muscles within our body. I wanted to thank you all for watching and taking the time for watching and taking the time, if you did, to answer the questions on the knowledge check. I hope you all are doing well.